The quest for the fountain of youth has captivated human imagination for millennia, but today's scientific community is approaching this ancient dream with unprecedented sophistication and evidence-based methodology. The emerging field of geroscience represents a revolutionary shift in how we conceptualize aging and disease, moving away from the traditional reactive approach of treating individual ailments toward a proactive strategy that targets the fundamental biological processes underlying age-related decline. The paradigm shift, from disease treatment to aging prevention. For generations, medical practice has operated under a compartmentalized model where each disease receives specialized attention and treatment protocols. This approach has yielded remarkable successes in managing specific conditions, yet it fundamentally misses the forest for the trees. Aging stands as the single most powerful risk factor for virtually every chronic disease that plagues modern society, yet until recently, it has been viewed as an inevitable natural process rather than a modifiable biological phenomenon. The traditional medical model treats cardiovascular disease with statins and blood pressure medications, addresses diabetes with glucose-lowering agents, and combats cancer with targeted therapies and chemotherapy. While these interventions have extended lives and improved quality of care, they operate within inherent limitations. Each approach addresses symptoms and consequences rather than the underlying driver that makes individuals susceptible to these conditions in the first place. This fragmented strategy becomes particularly problematic when considering the reality of modern aging. The vast majority of older adults do not suffer from single diseases but rather experience multiple concurrent conditions that interact in complex ways. A person might simultaneously manage diabetes, heart disease, arthritis, and cognitive decline, requiring a complex medication regimen that often produces conflicting effects and side effects. The traditional approach struggles to address this multi-morbidity effectively because it lacks a unifying framework for understanding why aging bodies become vulnerable to multiple diseases simultaneously. Geroscience offers a fundamentally different perspective by recognizing that aging itself represents a series of modifiable biological processes. Rather than waiting for diseases to manifest and then treating them individually, this approach seeks to intervene in the basic mechanisms that drive age-related vulnerability. The implications of this shift are profound, potentially transforming medicine from a reactive discipline focused on managing disease to a proactive field capable of preventing the onset of age-related conditions altogether. Understanding biological versus chronological age. One of the most important conceptual advances in geroscience involves distinguishing between chronological age and biological age. While chronological age simply measures the time elapsed since birth, biological age reflects the actual physiological state of the body and its deviation from what would be expected for someone of that chronological age. This distinction has practical implications that extend far beyond academic interest. Two individuals who are both 60 years old chronologically might have vastly different biological ages based on their overall health, fitness level, genetic factors, and lifestyle choices. One person might have the cardiovascular fitness, cognitive function, and cellular health typical of someone a decade younger, while another might show signs of accelerated aging that place their biological age closer to 70. The concept of biological age provides a more nuanced and accurate assessment of an individual's health status and mortality risk. Research has demonstrated that people with biological ages significantly older than their chronological ages face substantially higher risks of death and age-related diseases. Conversely, those whose biological age lags behind their chronological age enjoy protective benefits that extend both lifespan and healthspan. The measurement of biological age relies on various biomarkers and assessments that reflect the function of different organ systems. These might include cardiovascular fitness measures, cellular markers of aging, inflammatory indicators, and even epigenetic changes that occur in DNA over time. Advanced techniques now allow researchers to assess biological age through DNA methylation patterns, providing insights into the aging process at the molecular level. Perhaps most importantly, biological age appears to be modifiable through various interventions. Unlike chronological age, which progresses inexorably, biological age can potentially be slowed, altered, or even reversed through targeted approaches. This modifiability forms the foundation for therapeutic interventions designed to promote healthy aging and extend healthspan. The cellular foundation of aging. Understanding how to intervene in aging processes requires a deep appreciation of what happens to our cells and tissues over time. Aging occurs through multiple interconnected pathways that affect virtually every aspect of cellular function, from energy production to waste removal to genetic stability. At the molecular level, aging involves the gradual accumulation of damage and dysfunction across multiple cellular systems. DNA experiences increasing numbers of mutations and epigenetic changes that alter gene expression patterns. The protective caps on chromosomes, known as telomeres, progressively shorten with each cell division until they become so short that cells can no longer divide effectively. Cellular housekeeping mechanisms also deteriorate with age. The process of autophagy, which removes damaged proteins and cellular components, becomes less efficient over time. This leads to the accumulation of cellular debris that can interfere with normal function and contribute to age-related diseases. Similarly, the cellular powerhouses known as mitochondria become less efficient at producing energy and more prone to generating harmful reactive oxygen species. The maintenance of protein structure and function represents another critical aspect of cellular aging. As we age, our cells become less capable of properly folding proteins and removing those that have been damaged or misfolded. This protein dysfunction contributes to numerous age-related conditions, including neurodegenerative diseases like Alzheimer's and Parkinson's disease. Nutrient-sensing pathways also play crucial roles in aging. These cellular systems monitor the availability of nutrients and adjust cellular behavior accordingly. When these pathways become dysregulated with age, cells may inappropriately activate growth and division programs even when nutrients are scarce, contributing to cancer risk and other age-related problems. The stem cell populations that are responsible for repairing and regenerating tissues also decline with age. This reduction in regenerative capacity means that older adults recover more slowly from injuries and are less able to maintain optimal tissue function over time. Caloric restriction, the gold standard intervention. Among all the interventions studied in aging research, caloric restriction stands out as the most thoroughly investigated and consistently effective approach for extending both lifespan and healthspan across multiple species. The basic principle involves reducing total caloric intake while maintaining adequate nutrition, typically through a reduction of 20–30% compared to normal intake levels. The effects of caloric restriction on longevity have been documented across an impressive range of organisms, from simple yeast cells to complex mammals. In laboratory studies with mice, moderate caloric restriction has consistently produced substantial increases in lifespan, often extending median survival by 20–40% compared to control animals. These benefits appear to result from fundamental changes in cellular metabolism and stress resistance rather than simply from maintaining lower body weight. The CALERIE trial represented a landmark study in human aging research by providing the first controlled evidence that caloric restriction can influence aging processes in people. This carefully designed study followed participants for 2 years and documented significant changes in cellular aging markers among those practicing caloric restriction. Most remarkably, participants in the caloric restriction group appeared to age more slowly based on multiple biological measures, effectively gaining about 6 months of biological age advantage over the 2-year study period. The mechanisms underlying caloric restriction's anti-aging effects are complex and multifaceted. Caloric restriction appears to enhance cellular autophagy, improving the removal of damaged cellular components. It also upregulates DNA repair mechanisms while simultaneously reducing inflammatory responses that contribute to age-related tissue damage. These cellular changes translate into improved function across multiple organ systems and increased resistance to age-related diseases. Modern pharmaceutical approaches are now attempting to capture the benefits of caloric restriction without requiring the challenging lifestyle changes that traditional caloric restriction demands. Medications like semaglutide and tirzepatide, originally developed for diabetes treatment, produce substantial and sustained weight loss that may confer many of the same benefits as traditional caloric restriction while being much more practical for most people to maintain long-term. Metformin, the unexpected anti-aging candidate. Metformin represents one of the most intriguing examples of how existing medications might be repurposed for anti-aging applications. Originally developed and widely used as a first-line treatment for type 2 diabetes, metformin has attracted significant attention from aging researchers due to its effects on fundamental cellular pathways involved in aging. The drug's primary mechanism of action involves the inhibition of mitochondrial complex I, which triggers a cascade of cellular responses that may slow aging processes. This inhibition activates an important cellular energy sensor called AMPK, which in turn influences multiple downstream pathways involved in cellular maintenance and longevity. The activation of AMPK leads to the inhibition of mTOR, a central regulator of cellular growth and metabolism that plays key roles in aging. These cellular effects translate into enhanced autophagy and improved mitochondrial function, two processes that are central to healthy aging. By promoting the removal of damaged cellular components and supporting energy production, metformin may help maintain cellular health and function over time. Observational studies in human populations have provided compelling evidence for metformin's potential anti-aging effects. Large-scale analyses of veterans' health records have shown that people with diabetes who take metformin have lower rates of neurodegenerative diseases, including Alzheimer's disease, Parkinson's disease, and other forms of cognitive decline. These protective effects appear to extend beyond what would be expected from improved diabetes control alone. The COVID-19 pandemic provided an unexpected natural experiment that further highlighted metformin's potential benefits. Among hospitalized patients with diabetes, those taking metformin experienced lower mortality rates compared to those not taking the medication, suggesting that metformin may provide protective effects that extend beyond its primary indication. Despite these promising observations, researchers emphasize the need for carefully designed clinical trials specifically focused on aging-related outcomes. The existing evidence comes primarily from observational studies of people with diabetes, and it remains unclear whether metformin would provide similar benefits in healthy aging populations. Rapamycin, targeting cellular growth pathways. Rapamycin represents perhaps the most scientifically compelling anti-aging pharmaceutical currently under investigation. Originally developed as an immunosuppressive medication to prevent organ transplant rejection, rapamycin has demonstrated remarkable anti-aging effects across multiple animal models, consistently extending lifespan even when treatment begins relatively late in life. The drug works by inhibiting the mTOR pathway, a central cellular signaling system that regulates growth, metabolism, and aging. When mTOR activity is reduced, cells shift toward maintenance and repair modes rather than growth and division, potentially slowing the aging process and reducing susceptibility to age-related diseases. In laboratory studies, rapamycin has produced some of the most impressive longevity results ever documented in aging research. Studies in mice have shown lifespan extensions of 20–30% or more, with benefits observed even when treatment begins in middle age. These effects appear to result from improvements in multiple aspects of health, including reduced cancer incidence, improved cardiovascular function, and enhanced cognitive performance. The cellular mechanisms underlying rapamycin's anti-aging effects involve enhanced autophagy and improved cellular maintenance. By reducing mTOR activity, rapamycin promotes the removal of damaged cellular components and supports the cellular housekeeping processes that decline with age. This enhanced cellular maintenance may help preserve function across multiple organ systems and delay the onset of age-related diseases. Human studies with rapamycin remain limited but show promising initial results. Small clinical trials have demonstrated that short courses of low-dose rapamycin can improve immune function in older adults, as evidenced by enhanced responses to vaccination. These studies suggest that rapamycin's benefits might extend to humans, though much larger and longer-term studies will be needed to fully evaluate its potential as an anti-aging intervention. The challenge with rapamycin lies in balancing its potential benefits against known side effects. At the high doses used for immunosuppression in transplant patients, rapamycin can cause significant adverse effects, including increased infection risk and impaired wound healing. However, researchers believe that much lower, intermittent dosing regimens might provide anti-aging benefits while minimizing these risks. Senolytic drugs, clearing cellular zombies. Among the most innovative approaches in geroscience involves the development of senolytic drugs, which specifically target and eliminate senescent cells. These cells represent a unique cellular state where cells have stopped dividing but remain metabolically active, secreting inflammatory and harmful substances that contribute to aging and age-related diseases. Senescent cells accumulate throughout the body with age, and their presence correlates with numerous age-related health problems. These cellular zombies secrete a cocktail of inflammatory molecules, growth factors, and tissue-degrading enzymes collectively known as the senescence-associated secretory phenotype. This secretion creates a toxic tissue environment that promotes inflammation, impairs normal cellular function, and accelerates aging processes in surrounding healthy cells. The concept of selectively eliminating senescent cells while leaving healthy cells unharmed represents an elegant therapeutic approach. Early research in laboratory animals has produced remarkable results, with senolytic treatments extending lifespan by up to 27%, while improving multiple aspects of health including cancer resistance, cardiovascular function, and physical performance. The mechanisms by which senolytic drugs work involve exploiting specific vulnerabilities of senescent cells. These cells often rely heavily on anti-apoptotic pathways to survive despite their damaged state. Senolytic drugs target these survival pathways, essentially forcing senescent cells to undergo programmed cell death while leaving healthy cells unaffected. Human clinical trials with senolytic drugs are in early stages but show promising initial results. These studies have demonstrated that senolytic treatments can safely reduce the number of senescent cells in human tissues and may improve various measures of health and function. However, much larger and longer-term studies will be needed to determine whether eliminating senescent cells can meaningfully improve healthspan and lifespan in humans. The development of effective senolytic drugs faces several challenges, including the need to identify compounds that can effectively target senescent cells while avoiding harm to healthy tissues. Additionally, researchers are working to understand the optimal timing and frequency of senolytic treatments, as completely eliminating all senescent cells might interfere with normal physiological processes where these cells play beneficial roles. Regulatory and implementation challenges. Despite the scientific promise of anti-aging interventions, significant regulatory and practical challenges stand in the way of translating research findings into clinical applications. The most fundamental obstacle involves the fact that regulatory agencies like the FDA do not currently recognize aging itself as a disease that can be prevented or treated. This regulatory framework creates a paradoxical situation where researchers must design clinical trials around specific age-related diseases rather than aging processes themselves. Instead of directly testing whether an intervention slows aging, researchers must demonstrate effects on traditional disease endpoints like heart attacks, strokes, or cancer incidence. This approach significantly complicates study design and may miss important benefits that don't fit neatly into existing disease categories. The challenge becomes particularly acute when considering that many potential anti-aging interventions might require years or even decades to show their full effects. Traditional clinical trial designs are poorly suited to evaluating interventions that work gradually over long time periods, especially when the endpoints involve preventing diseases that might not occur for many years. Additionally, the development of anti-aging therapies requires new approaches to dosing and administration that differ significantly from traditional disease treatment models. Many promising anti-aging interventions appear to work best at lower doses or with intermittent dosing regimens that differ from standard therapeutic approaches. These alternative dosing strategies require extensive safety and efficacy testing that current regulatory frameworks are not well equipped to evaluate. The cost and complexity of conducting large-scale, long-term studies of anti-aging interventions also present significant barriers. Such studies would need to enroll thousands of participants and follow them for many years to detect meaningful effects on aging processes and age-related diseases. The financial and logistical challenges of conducting such studies are substantial and may require new models of research funding and collaboration. Future directions and implications. The field of geroscience stands at an exciting crossroads where decades of basic research are beginning to yield practical interventions that could transform how we approach aging and age-related disease. The convergence of our understanding of aging mechanisms with the development of targeted therapeutic approaches creates unprecedented opportunities for extending healthy human lifespan. The implications of successful anti-aging interventions extend far beyond individual health benefits. In an era of rapidly aging populations worldwide, therapies that can extend healthspan could have profound economic and social impacts. Reducing the burden of age-related diseases could dramatically decrease healthcare costs while allowing older adults to remain productive and independent for longer periods. However, the successful translation of anti-aging research into clinical practice will require continued innovation in research design, regulatory approaches, and healthcare delivery models. We will need new ways of measuring and evaluating aging processes, novel clinical trial designs that can capture long-term benefits, and healthcare systems that can effectively integrate preventive anti-aging interventions into routine care. The journey from laboratory discoveries to clinical applications remains long and uncertain, but the potential rewards justify continued investment in research efforts. As our understanding of aging mechanisms deepens and our therapeutic tools become more sophisticated, the prospect of significantly extending human health span moves from the realm of science fiction toward achievable reality. The ultimate goal is not simply to add years to life, but to add healthy, productive, and fulfilling years. By targeting the fundamental biological processes that drive aging, we may finally achieve the ancient human dream of aging gracefully while maintaining vitality and independence throughout our extended lifespans. This represents not just a medical advance, but a transformation in how we conceive of aging, from an inevitable decline toward a manageable biological process that can be optimized for health and longevity.